Good evening, everyone. I am Jyoti. I am practicing in Jesus College of Arts, Commerce and Science in Mysore. Today I am here to present my seminar topic Ion Exchange Chromatography. Before going to explain my topic, firstly let me teach you about chromatography. Chromatography is a physical method of separation of a mixture of substance into its individual components by distributing them between two phases like a stationary phase and a mobile phase. There are several different types in chromatography. Based on separation process, today I am going to explain ion exchange chromatography. Ion exchange chromatography is based on a phenomena of attraction between opposite charges. The stationary phase is selected a particular charge and attracting the sample molecule with opposite charge. The stationary phase is also known as an ion exchanger. Ion exchange chromatography is widely used for separation and purification of proteins, peptides, nucleic acids, polynucleotides, etc. In ion exchange chromatography, there are two types: cation exchanger and anion exchanger. Cation exchanger as a negatively charged that attracts a positively charged cation. So while anion exchanger is positively charged and attracts negatively charged anion. Different functional groups such as carbonyl, carboxymethyl, etc. can be used as a cation exchanger. Before having a negative charge ions like acetate, phosphate, etc. can be used with cation exchanger. On other hand, functional group like aminoethyl, trimethyl aminoethyl, etc. can be used as ion exchanger. The buffer used with anion exchanger as a positive charge, for example risperidine, pyridine, etc. Next is selection of ion exchange resin. Depends on the first one is type of ion to be separated, cations or anions. Second one is nature of the ions to be separated, like strong or weak. Third one is efficiency of the resin measured by ion exchange capacity. Here ion exchange capacity is the total ion exchange capacity in terms of the exchangeable functional groups expressed as mg/gram of the ion exchange resin. The fourth one is particle size of the resin, like 50 to 100 mesh or 100 to 200. Fifth one is structural type of the resin, like pores, pellicular, etc. The sixth one is amount of cross-linking agent present, which decides the swelling of the resin. Next is common ion exchange resins. Common ion exchange resins we can see as is shown in table, like a class of resins: cation strong, cation weak, their nature, pH range and applications. We can see that in as I following as I show in table. Next is cation and anion exchange. In case of cation exchange, the stationary phase is positive charge on it. When mixture pour into the column, the stationary phase attracts a positively charged molecule. Other molecules are washed away. Similarly, in case of anion exchange, the ion exchanger is positively charged on it. When mixture pour into the column, the stationary phase attracts negatively charged molecule, the cations, from the mixture. Other molecules are washed away from the column. Now to extract the target or bonded molecule, the pH or ionic strength must be changed. To achieve it, we had a buffer of different pH or ionic exchange into the column. When pH change, the target molecule separates from the stationary phase and free out of the column. Next is factors affecting ion exchange separations. First factor is nature and properties of ion exchange resins, cross-linking and swelling. Second one is nature of exchanging ions, valency of ions at low concentration and ordinary temperature. Extent of exchange increases with increasing in valency as I show in following order. Next is applications of ion exchange chromatography. Ion exchange chromatography is a brilliant technique to extract ion and charged molecule from a mixture. It is mainly used to extract proteins, sugars, and amino acids from a mixture and to isolate the inorganic ions from a solution and to kick out of ionic impurities from a solution and for softening of odd water and to isolate enzymes from a mixture, etc. Thank you.